Hey guys, welcome back to One Stop Biology. And right now we are at the second chapter of class 12th NCERT Biology. So right now we are dealing with sexual reproduction in flowering plants, right? So till now what we have studied is pre-fertilization events, including how microsporogenesis occurs, how megasporogenesis occurs, and then in the last video we saw how does pollination occurs. So when it comes to pollination, it's basically like what are the different types of pollination, what are the different agents of pollination, right? And what is the the most comfortable maybe type of pollination for different type of plants, right? So today we are going to start with pollen and pistil interaction after pollination which occurs, and then we will also study double fertilization, right? So now, pollination itself basically does not guarantee the transfer of right type of pollen, right? So, by right type of pollen, I mean that the pollen should be compatible and it should be of the same species as the stigma on which it is going to fall, right? So, pollination does not guarantee that the right type of pollen is transferred to the right type of stigma, right? So what happens several times, very often this happens that pollen of the wrong type, okay, so either from other species or from the same plant that lands on stigma. And then what happens is that the pistil, right? So the female part has the ability to recognize the pollen. So whether it is of the right type or it is of the wrong type. So whether it is compatible or it is incompatible. Now, if it is of the right type, the pistil accepts the pollen and it promotes post-pollination events, which is the interaction between pollen and pistil. But if the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination or fertilization. Okay, so the ability of the pistil to recognize the pollen followed by its acceptance or rejection is nothing but the result of a continuous dialogue components of the between the pollen grain and the pistil. So remember, right? Now this kind of reaction is basically happening between chemical components of pollen and that of pistil. So the chemical components of pollen and pistil basically recognize each other in case of compatible pollen to pistil, right? So now what happens is once the compatible pollination occurs, so once compatible pollination occurs, what happens is the pollen grain germinates on the stigma of the pistil, and it produces the pollen grain produces a pollen tube through one of the germ pores. Now remember that I have shown you in the diagram of a germ pore in the previous video. So if you have not gone through the previous video, please go through it first, okay? So if we see this diagram, if we see this particular diagram, what we are looking at is the pollen tube germinating from the, so this is the pollen tube and it is germinating from the pollen grain, right? So what happens is after compatible pollination, the pollen grain germinates on the stigma. So this is the stigma, right? So it germinates on the stigma to produce a pollen tube through one of the germ pores. Now what happens? The contents, so whatever is inside the pollen grain, it moves into the pollen tube, right? And pollen tube keeps growing through the tissues of the stigma and style, and it reaches the ovary. So here if you see that this pollen tube is reaching to the ovary here, right? Now you would also remember that previously we have studied that pollen grains can be shed either in two-celled stage or three-celled stage, right? So in two-celled stage, one was generative and one was vegetative cell, right? And in the three-cell stage, we have one vegetative and two generative cells, right? So in two-celled case, in the case of two-celled stage, the generative cell, So one is generative cell, the other one is vegetative cell. So in two-celled case, the generative cell, what happens is it divides and forms two male gametes. Okay, so they form two male gametes during the growth of pollen tube in the stigma. Now, what happens in three-celled stage? So, in three-celled stage, pollen tubes carry the two male gametes from the beginning. Okay, now pollen tube after reaching the ovary, it enters the ovule through the micropyle and then enters one of the synergids through the filiform apparatus. Okay, so we also studied the structure of the entire egg apparatus, right? We studied the structure of the entire embryo sac. So we studied where exactly in the synergid is the filiform apparatus, and there we studied that it guides the pollen tube when it enters inside the synergids, right? So basically, that synergids that guides the entry of the pollen tube, right? Now, all these events from pollen deposition to the stigma until pollen tube entering the ovule are basically together known as pollen-pistil interaction. Okay. Now, this pollen-pistil interaction is a dynamic process and it involves pollen recognition followed by promotion or inhibition of the pollen and then formation of the pollen tube which enters inside the embryo sac, right? So, if we now study this diagram, this is nothing but the pollen tube. These are the antipodal cells. This is the polar nuclei. Here we have one egg cell and we have synergid, right? So this is the exact diagram which you'll have to remember. Okay. Now, if we also see the next diagrams, okay, so here if you see the entering of the pollen tube, you'll see that so basically this is nothing but the central cell which has two polar nuclei, right? This is the egg nucleus. Here we have one synergid. This is nothing but the plasma membrane. Then we have filiform apparatus. Here we have the two male gametes and vegetative cell. So vegetative nucleus. Okay. And now if you see, so basically this is an egg apparatus which is showing entry of pollen tube here into a synergid. And this is nothing but discharge of male gametes into the synergid. So these are the male gametes. These are the polar nuclei. So this is discharge of male gamete into a synergid and movement of these sperms. One is moving towards the sperm and the other one is moving towards the center cell, which is basically where we have the polar nuclei, right? So this is the next diagram. I hope you understood this diagram well. If not, please do let me know in the comment section and I'll explain it again. Okay, so this is basically the pollen pistil interaction. So the next topic is artificial hybridization. So artificial hybridization is one of the major approaches in case of crop improvement programs. So in such cases, what happens is the desired pollen grain are used for pollination and stigma is before that stigma is protected from contamination. So basically stigma is protected from any unwanted pollen. And this is achieved by emasculation and bagging techniques. Okay, so what happens is emasculated flowers, they are covered with a bag of suitable size. Okay, and these bags are usually made of butter paper, and it prevents contamination of its stigma with unwanted pollen. And this process is nothing but bagging. Okay, so when the stigma of bagged flower attains receptivity, mature pollens are collected from anther of the male parent and it is dusted on the stigma, and the flowers are again rebagged, and then the fruits are allowed to develop. Right? So if the female parent produces unisexual flowers, there is no need for emasculation, right? Because any which way there are no flowers closer which will basically contaminate the stigma, right? The female flower buds in that case are bagged before the flower opens, and when the stigma becomes receptive, pollination is carried out using a desired pollen, and the flowers are rebagged. Remember, okay? So this is nothing but a technique of artificial hybridization. Now let's move on to double fertilization. So after the pollen-pistil interaction happens, after the pollen tube enters into one of the synergids, the pollen tube does what? It releases the two male gametes into cytoplasm of synergid. Okay. Now one of these male gametes move towards egg cell, right? And fuses with its nucleus, thus completing the fertilization or syngamy. Okay, so this results into a diploid zygote, a diploid cell zygote. Now, what happens to the other male gamete? So, the other male gamete moves towards the two polar nuclei which are located in the central cell. Okay, so here if you see this diagram, this is the central cell. This is nothing but the center cell. Here you have the polar nuclei. Okay, so the other male gamete, it moves towards the two polar nuclei say located in the central cell, and it fuses with them and produces a triploid, because there are two polar nuclei already, right? So it's already two-celled, and then again one cell of male gamete forms a triploid primary endosperm nucleus, which is also known as pen. Okay, so this involves fusion of three haploid nuclei, and that is why it is known as triple fusion. Okay, now since there are two types of fusion, one is syngamy, which happens between the egg cell and one of the male gamete, which is diploid. So one is syngamy and the other one is triple fusion. This phenomenon is basically known as double fertilization. Okay, which is an event very unique to flowering plants. Remember that double fertilization is a very unique event which happens in flowering plants. Now the central cell where triple fusion has occurred becomes the primary endosperm cell peck. Okay, and that develops into an endosperm, and the zygote which has developed from syngamy, that develops into an embryo. So now if I have to label this diagram, how will I do that? So here if you see, synergids are degenerating, so these are degenerating synergids. Okay, then here you have zygote. So syngamy has occurred and this is diploid zygote. Then you have PEC, primary endosperm cell, and here you have primary endosperm nucleus, which is 3N. And this is nothing but double fertilization. And here you have antipodal cells, right? Again, these are degenerating antipodal cells. Okay, so it's doing what? Basically, these degenerating cells are helping in nourishment of the other parts of the embryo sac. So it helps in nourishment basically. Okay, so now let's move on to the next diagram. Okay, so this basically helps in triple fusion, and this is nothing but double fertilization, which is done between two types of cells. One forms the zygote and the other forms the endosperm, the primary endosperm cell. And these form nothing but the endosperm and the embryo. Okay. Now with this we have finished double fertilization. In the next video, we are going to cover post-fertilization events, which is now the formation of endosperm, the formation of embryo, and then from that, the formation of fruit and seed. So I hope you understood both the pollen-pistil interaction and double fertilization very well. Guys, if you have any doubt as such, please don't hesitate in sending your doubts either on the WhatsApp number that I've provided in the description of the video or over the comment on the video, and I'll clear your doubts. And if you did understood it, please do not forget to like the video and share it with your friends. And also please do not forget to subscribe to the channel. Thanks guys. Bye bye.